The overall goal of this methodology is to create macroporous hydrogels with varying pore sizes for cell culture applications.
This method can help answer key questions in the field of 3D cell culture, such as the principal feasibility of a novel hydrogel material for 3D cell culture applications.
The main advantage of this methodology is that it offers a range of techniques to optimize and adjust a novel material to the requirements of different types of cells.
To begin, dilute 1 mg of the RGD peptide or an equivalent cell-adhesive peptide in 100 microliters of sterile water to obtain a 10 mg per milliliter solution. Next, remove the bottom of a 96 well plate and replace it with removable plastic wrap. Mix 100 microliters of BSA stock solution and 100 microliters of THPC stock solution in the 96 well plate to obtain 200 microliters of the hydrogel. Mix the components by pipetting up and down at least 5 times to guarantee a uniform hydrogel after polymerization. Place the 96 well plate at room temperature for about 10 minutes until all hydrogels are properly polymerized. Following polymerization, carefully and slowly remove the plastic wrap from the bottom of the plate. Press the hydrogels out of the 96 well plate using a small stamp and transfer them to 1.5 milliliter tubes with sterile PBS pH 7.4. Store the hydrogels in phosphate buffered saline at 4 degrees Celsius for up to several months. To freeze dry